we have today, we had today 2 talks that were very special on the session of 25 years of PDK1 Akt, and now we have yet another very special presentation. All the rest will have the format of shorter talks, but this is also a very special one which will also be recorded and hopefully be available for the whole community for the future as well.
Okay.
So this presentation today, it's a great pleasure for me to present Susan Taylor. Susan Taylor to this audience does not really need an introduction, so I will not mention all the awards, but I would mention, for example, that she is a member of the National Academy of Sciences already since 1997. What I will not introduce is her CV, but I would like to introduce what her impact has been on myself and all of us here today. So, because she's well known because of her publications, but it's perhaps most known by the impact he has had on all of us, not only from the publications, but also from the discussions that she has had throughout her career. She's always been very open, discussing and giving always very valuable advice to many people over the years. And this type of impact is sometimes not so easy to visualize, because it just dilutes in the general population and, for example, leads Dario to believe that I had an important participation in some discoveries, whereas from my perspective, it all I owe to Susan and her previous work. So I would like to myself give my vision on the impact he has had on myself and on, I think, all of us today through my personal experience. So when I went to Institut Pasteur in Paris in 1996 as a postdoc, but before I had been as a student, the people in the laboratory, they were discussing about how the catalytic domain of protein kinases and how the catalytic domain of protein kinase A needed to be complemented. On its own, the catalytic domain was not a kinase, a functional kinase. That was a suggestion, but it needed to be complemented, for example, by an N-terminal extension attaching to the site, and by a C-terminal extension with a hydrophobic motif binding to a particular site on the small lobe. And the discussion was there, and it was not just this discussion was there because of the discussions with Susan Taylor. The head of the lab had published a paper already in 1994 with Susan. Other members of the laboratory in Paris, like Miguel van Bemmelen, he had spent 6 months in Susan Taylor's lab before joining a Pasteur lab where I was. So the influence that she had on this work that we've done into the AGC kinases regulation is exactly what she had been, or what was being discussed at the time in Paris. The complementation of the catalytic domain, we followed with the small molecules, but it's the mechanism of AGC kinase regulation by phosphorylation and so on. So I just to let you know the impact that we know and the impact that sometimes we don't know when it dilutes into the huge impact that Susan has had over the science and ourselves. So it's a great, great pleasure, Susan, to have you here today and to give this very special talk for this meeting. Thank you.
Thank you, Ricardo. And it's really great to be here. I thought, oh, I'm crazy to come for just like 4 or 5 days to Europe, but I've already learned enough to make it worthwhile for having come. So I'm going to go back in history at least 15 years or even more from what Dario told you. But the very beginning was like 60 years ago with Ed Krebs and Eddie Fisher with that first phosphorylase kinase. And then PKA was second. So the second one to be discovered a decade later. And then we had all this activity of tyrosine kinases and tyrosine phosphorylation with Tony. But Tony and I came to San Diego about the same time in the early 1970s, neither one of us working on protein kinases, but by the end of that decade, we were all part of this family. And so I'm going to focus on PKA, what we've learned from it, what we missed. And then, in 1991, we got the first structure. So that was over 30 years ago now. So, what did we learn and what did we miss? And then if I have time at the end, I'll do just a tiny bit about LRRK2. And I want to also point out that right after PKA was discovered, 1972, PKI was discovered, 1971. It's a pseudosubstrate. It's an intrinsically disordered protein, although we didn't really even know what that was then. And it had this inhibitory sequence at its N-terminus. And that was characterized very well in the 1980s. And then later on, we discovered that there's another motif there, a nuclear export signal. That was the first discovery of a nuclear export signal. So this is an intrinsically disordered protein, has two important binding motifs, small linear motifs. And I think its role in cell biology still really needs to be elucidated. I think that's not, but it's a pseudosubstrate. It has an alanine instead of a serine or a threonine. And so what were we doing in those early days when I first started working on it? I knew how to run columns and do peptide mapping. My first graduate student, Mark Zoller, was using an analog ATP mimetic to identify active site residues. And he used this FBISBA to identify one specific lysine in the catalytic subunit that was modified. It lost activity, and you could protect with ATP. And so we thought this is at the active site. And then another graduate student cross-linked with carbodiamides and found two carboxylic acids that could cross-link. So we had kind of this triad. And again, just historically, PKA was the first protein kinase to be sequenced by the group in Seattle in 1981. SARC had been cloned in 1979 and had been predicted to be a protein kinase. But in 1979, nobody knew what a kinase looked like. And so when the first sequence came out from Seattle, they didn't recognize it, but Margaret Jayhoff did. She was a pioneer bioinformatics person, and she could align it. It was not a lot of sequence homology, but she could align these motifs and showed that SARC came from the same gene family as PKA. And so now all of a sudden you had a very big kinome that included not only all those metabolic kinases involved in energy, but also the oncogene kinases at the top. And so, and the P site, again, we isolated these also by all those columns that Dario talked about with our laborious protein purifications. And so this was enough in 1988 for Tony and Steve Hanks to align them and find these motifs. So there's like these are still a good indicator of secondary structure. But to me, it's kind of like a Rosetta Stone. What was that lysine 72 doing? It's conserved in all of these kinases. And so that was the first structure in 1991. And, what we've learned since then is that dynamics is really, really important in the function of all of these protein kinases. And we didn't really appreciate it at the time we did that first structure. And so we saw the first structure. It told us that fold is going to be conserved in all kinases, that the little white dots, those are the conserved residues. That's lysine 72. Mostly they cluster around the active site. They were going to be conserved. ATP binding, it's in that pocket there. Peptide binding, this was an inhibitor peptide that this first 20 residue peptide from PKI that binds with like a close to nanomolar affinity. So we were really lucky with this structure. And it's actually two structures, 1991 and 1993. And I'm going to point out while this is fully phosphorylated, PKA, unlike many of the kinases we've been talking about, it's activated, it's synthesized as a fully phosphorylated protein and then stored as an active kinase, so it can be reversibly and rapidly activated in response to second messengers, like in this case cyclic AMP. And so if we look at, there's that little triad that we identified by our affinity labeling. So that was the first thing that we looked for, or that I looked for. So this is, we call it a regulatory triad that's at the active site. So that must be assembled before you get any kind of phosphotransfer, I think. And then the phosphate on the activation loop, you know, it's doing much more than just an electrostatic node. It's really integrating all parts of the kinase. And you have, like, this is the only charge group that comes from the N lobe. The rest are all from the C lobe. And it really is only fully active when you have that phosphate there. So it's really playing an important role in integrating the whole active structure. As I said, that's the only one that comes from the small lobe. And so what we didn't emphasize at the time is that this was a different kind of nucleotide binding motif. It's a G loop, and it's not a P loop like ATPases, and now we know GTPases. They have this P loop. And it's a fundamental difference of how this phosphate is buried under the beta strands here. This adenine ring is buried under the beta strands here. The phosphate is buried under the glycine-rich loop here. And I'll come back at the end to point out again the difference between a G loop and a P loop. It's a fundamentally different mechanism. So I wanted to tell you something about that first structure, which maybe many of you don't appreciate. The first structure in 1991 was actually a binary structure. It had PKA and it had the IP20 peptide bound to it. And there were two back-to-back papers. One was the structure of the kinase fold. The other was a detailed description of how this IP20 peptide bound to the kinase, because there'd been so much work done by Don Walsh and John Scott and others, especially Don Walsh, of mapping all those sites, the hydrophobic motif, the importance of the hydrophobic residues. Again, this is a pseudosubstrate. It's not a substrate. It has an ALA instead of a serine. We knew from that first structure ATP was in the buffer. You could see from omit maps that it should be at the cleft of the between the two lobes, but we couldn't get a good resolution of that. And it wasn't until the 1993 paper where we now saw the really well-resolved ATP. And this one here now had IP20C, ATP, and it had excess magnesium. So that magnesium, we had substoichiometric magnesium, 1 millimolar ATP and 0.5 millimolar magnesium, and you did not see the ATP without that excess of magnesium. And so I think if you look at the binding in the ternary complex of IP20, it's about 1 nanomolar. ATP to that complex is about 60 nanomolar. So it's really binding much better than 25 micromolar, which is the KM. So I think we had a pseudosubstrate, we had ATP binding with high affinity, and so we trapped this very stable complex. This is how we presented the ATP binding site back then. A lot of the electrostatic interactions, in this second one, the two magnesium ions, but if you look at the hydrophobic residues, they're as conserved as the lysine. This K72 is conserved in all kinases. Those hydrophobic residues are absolutely conserved, the A and the V and the F as well in the glycine-rich loop. And then a couple other hydrophobic residues. And so we didn't really appreciate the importance of the hydrophobic residues for really binding, especially the adenine ring, and at the edge, the phenylalanine in the glycine-rich loop. So the importance of the hydrophobic residues we really came to appreciate much later. And so then in the next several years, we saw several structures, and we also realized that, more about this C-terminal tail, that there's a very stable part of it here, and then here's a little hydrophobic motif that we've been hearing about. But this part right here is very dynamic. In the apo, this is really disordered. You can't see it. But you can see ADP bound, and then when you have it fully closed, it's only really fully closed when you have ATP and 2 magnesium ions. And this C-terminal tail is highly dynamic. We didn't appreciate that at all from the first structure. And the glycine loop is highly dynamic. And you can, one of the other experiments from those early days was another graduate student, Adam Bastidas, and he wanted to actually look at the myrcellation site, and he wanted to get this CPKI structure. And he went to look for IP20. We didn't have IP20, but he had SP20. It was in the freezer. So he crystallized it. And he crystallized it with AMP, PMP, so it wouldn't hydrolyze. And his structure, this was the structure that he got. And initially he did see the substrates. He saw the SP20 and AMP, PMP. But that was only like 45% of what he saw. He saw a second crystal there, and these were the products. So he actually had trapped in that crystal both the substrates and the product of the ADP analog and the phosphorylated substrate. And so, I mean, that was pretty remarkable. And then he went and he let that go. That was after 2 months. And then after 6 months, he found the product. But he also then could find that one of the magnesium ions, the density for one of the magnesium ions had disappeared. And so he could actually map with these early experiments the whole catalytic cycle for this substrate. This substrate, because it has that amphipathic helix, it binds tighter than chemtide. You wouldn't get a little chemtide kind of peptide sticking around, but this one did, and so it allowed Adam to actually trap all of the steps of catalysis in a crystal lattice. And one of the things that was most important was this linchpin magnesium ion that is needed in order to get a tight complex to form in the first place, and you have to remove that one of the magnesium ions before you can remove ADP. And that was predicted by Andy McCammon computationally back in 2009. So this is, see if I can make it work. You can see it going from an open conformation, ADP, to a closed conformation. You could see how the C helix is moving a little bit, and you could see how the ATP and the peptide then go when it's in a fully closed conformation like that. So it's this motion of the glycine loop and dragging down the C helix as well, with the hydrophobic motif really anchored to the C helix here. And I can do that one more time. So I think you can see those motions, and you can see the red part. That's the C-terminal tail wrapping around the end lobe. And I'll talk about that a lot more tomorrow. But this is the conserved kinase core. So you can map any active kinase from this structure of PKAs. 1 ATP is the PDB number. And AlphaFold gives you exactly the same thing, this fully extended activation loop that we see there. But that by itself is not active, and so it's really necessary to have those tails. There's the tails that wrap around both the end lobe and the C lobe. And here's this C-terminal tail highlighted here. And so we call this one the active site tether, that middle part that I told you is dynamic of the C-terminal tail. There's 2 residues in that tail, a phenylalanine and a tyrosine. And what Kanan Natarajan did when he was a postdoc in my lab, he showed that this tail, this C-terminal tail, is conserved in all the AGC kinases. And so I didn't realize how unusual that is, but it is very unusual, and I'll try and point out to you why. So you look at here, when you look at a close-up of that ATP binding site, there's 2 residues, this phenylalanine and this tyrosine, that are really part of this, and they're really shielding the adenine ring, the ribose and the adenine ring here. And if you knock those out, the kinase is not active. These are not in the kinase core. They are in that C-terminal tail. So this is very unusual, and I didn't really appreciate just how unusual it is. Here you can see from Kanan's study, this is like the core, which is pretty much the same in all of them. And then you can see, now you melt away the core, and you can see the tails of all those AGC kinases. And that this tail, this is conserved, it's pretty much folded the same. Here from about, there's a PXXP after that. This is the part that's dynamic in catalysis. And here's the hydrophobic motif, which is also very stable. Processing of that tail is unique, as you all know, for every kinase, and essential and very complex. And it involves heterologous kinases as well as autophosphorylation in some cases. So how do the AGC kinases differ from other kinases? Or maybe just this is PKA and PKC, that they are assembled as fully active kinases, and then they need to be activated quickly and reversibly by second messengers. And I think from what we've heard, IP3, maybe it's just PKA and PKC that are like this, but they respond to both cyclic AMP and calcium oscillation. So they're made as a fully active kinase. Activation loop is phosphorylated, and it really says why we were so lucky with that first structure. So between the next 30 years or so, we did a lot of different things, and I'm not going to go through any of those looking at all the properties, but just point out the hydrophobic regions. There's 2 spines that we showed. This is what we call the catalytic spine, and this is the regulatory spine. This is filled by the adenine ring, completed and joined the N lobe and the C lobe. And the red one is the regulatory spine, which is assembled in a very dynamic way. So this C spine here, it's just filled by adenine ring. If you have the adenine ring, now you link the hydrophobic architecture of the N lobe with this extended hydrophobic architecture of the C lobe. Includes this very hydrophobic F helix. It should be a transmembrane helix. You don't usually find a hydrophobic helix buried in the middle of a globular protein like this. And then our spine is these 4 regulatory, they're not contiguous, but they're all assembled. It's a hallmark signature along with those 3 charges of every active kinase. And then this just shows you, like SARC, a non-AGC kinase, how it's activated. And it's made as an inactive kinase. You can see the spine is broken. And now when it's activated, you can see how the spine is assembled. And if you look carefully, you can also see how this little triad here, I can do it again, this little triad comes together and the activation loop goes out. So if I do it one more time, this is a non-AGC kinase, and that assembly of the regulatory spine happens once you activate it. So there really is a fundamental difference. Oops. How is PKA regulated, and how do we achieve specificity? So this is now a lot of Nobel Prizes on this slide here, but this is the receptor activated in response to hormone binding or neurotransmitter binding. And then you have the whole G protein coupled activation, which wasn't discovered until the mid-1970s. And then you make cyclic AMP. And so cyclic AMP, its primary target, not its only one, its primary one in cells, is the regulatory subunit, which is assembled as a holoenzyme. And it's also bound to A kinase anchoring proteins. I'm not going to say too much about those now, but it targets it to specific sites in the cell. And the activity of cyclic AMP is unleashed when you bind cyclic AMP to the regulatory subunits. It doesn't mean it has to completely dissociate, but the activity is unleashed. And if we think about it, we started obviously with minimalization of the C subunit and the R subunit, but in cells, it's really that tetramer, and it's really a dimer of dimers. The R subunit is a dimer with a dimerization domain at the N-terminus. There are 4 holoenzymes. They're functionally non-redundant, so they're doing different things. They localize differently in the cell. And then they are bound to ACAPs. So there are lots of ACAPs. Some are small, but most are very big and have many binding motifs. And they assemble it and target it to specific sites in the cell. And so I think of these like PKA signaling islands. And there's lots of PKA signaling islands in the cell, and they probably are each committed to their own channel, receptor, transporter. They're not exchanging. That's their function if you look at physiological PKA signaling. Okay, so I'm going to tell you very little bit about the regulatory subunits, just that they're ordered very differently. The N-terminus is this dimerization domain, which I told you about already. This is the ACAP peptide. It's an amphipathic helix again. And you have 2 cyclic AMP binding domains, but they're linked by this intrinsically disordered region that is disordered in the R subunits. It becomes ordered when it binds to the C subunit. So I think it's really dynamically ordered segments. And these are very typical in many signaling proteins. And so we did like the first R subunit, and then you could see the cyclic AMP binding domains, but the little inhibitor site was not visible here in this structure, just the 2 cyclic AMP binding domains. And it wasn't until we went to RC complexes that you could really understand. This is the inhibitor site here, and how the regulatory subunit docked that inhibitor peptide into the active site cleft and really kept it turned off so that it's cyclic AMP binding to the cyclic AMP binding site that activates this kinase. And but if you looked at this, the RC complexes like this, they all look more or less the same. So you couldn't understand the functional diversity until you went to the holoenzymes. And these are also functionally distinct. If you knock this out, you see learning defects, you see different diseases associated with this. If you knock out R1 beta, well, R1 alpha is more endocrine diseases. If you knock out R1 beta, they're learning defects. And you can, some of these mutants, now there are only a few, you can see fibers in the brain. They're very different phenotype. This one is the one, I think it's like a metabolic sensor, an energy sensor. It's the only one that's embryonically lethal. It's a major sensor of stress, different kinds of stress, and it's also the tissue-specific extinguisher of cyclic AMP-mediated gene expression. And nobody's really explained that fully, so I think there's still a lot of questions outstanding. And this is looking at mutations. There's lots of single-site mutations as well as many that induce nonsense-mediated decay. Even if you have half as much R1 alpha, you have a phenotype. And so the ones that are easier to activate are localized mostly to the first cyclic AMP binding domain. Those that are harder to activate are localized to the second one. And cardiac complex disease is heart defects and endocrine defects, gigantism associated with cardiac complex, and skeletal defects and dwarfism are associated with the ones that are harder to activate. So it's very finely tuned. 10 or 20% difference gives you a phenotype. And just one last piece of information, that all of them are the same organization, the 4 R subunits, but there's a fundamental difference. This is like the R2 subunits. Most R2 subunits are permanently anchored to different targeting sites, channels, receptors, transporters. And they're bound very tightly, 1 to 5 nanomolar, so they stay there. They really are tightly anchored. The R1 subunits have it's an alanine. The arrow should be over. It's an alanine or a glycine at the P site. They are pseudosubstrates, whereas the R subunits are substrates. And so that's a really fundamental difference when you look at the function of these two. So we have 2 pseudosubstrates, PKI, and all of the R1 subunits are pseudosubstrates. And I would like to know from you, are there other kinases that have pseudosubstrates that play a major physiological role in regulation for any other kinases, but any of the AGC kinases? Because these are really important pseudosubstrates. And here's the RC complex of an R2 subunit, and you can see this little beta 4, beta 5 loop is solvent exposed. This is like what we call FDDY motif. It's in the C-terminal tail, and this C-terminal tail is actually pretty disordered when you just look at this RC complex. But if you look at the holoenzyme, I think then you can see why it's so important to see the holoenzyme, because then you see, for the first time, you see the symmetry, and you see now these regions that were solvent exposed in the free C sub in the RC complex now provide the dimer interface, a dimer of dimers. And again, it's pointing out this is going to that C-terminal tail, to that disordered part of the C-terminal tail. So that disordered part of the C-terminal tail is really sensitized to making protein-protein interfaces, and it's different for R1, it's different for R2. There are other things that can go to that little tail as well. And I'll talk some more about that tomorrow, but I think just why it's so important to see the full-length protein. And here's just, when we crystallize it, you don't need ATP. You need ATP to really get a nice stable complex of R1. You don't need it for R2. But what you can see here is that you have a beautifully formed chamber for binding ATP. You have this C-terminal tail is perfectly ordered, and it's ordered by the beta 4, beta 5 loop coming from the other subunit, from the other R subunit, not its own R subunit. So it's perfectly forming that ATP binding site. You couldn't do that in PKI or R1 alpha without having ATP there. And so Ping, who did this structure, said, okay, what happens if you add ATP? And I said, oh, it'll just dissociate, but it didn't dissociate. it transferred the phosphate. And so now you had a stable complex with ADP and the phosphorylated inhibitor site. And it takes cyclic AMP to make it dissociate. You can see this is like a chamber. You can follow the phosphorylation in this chamber without dissociating the R subunit. And it's only this little bit at the inhibitor site that becomes more loosely associated. Yeah. Oh, okay. So they're all different. The quaternary structures are different, and probably there's different quaternary structures of like R1 alpha depending on the energy state of the cell. So the quaternary structures are really important, let alone the ACAP anchored structures, which are also going to be important. And so these two, R1 beta and R2 beta, are highly expressed in the brain. And so we asked this question, how are they localized? Because like Roger Chen was always saying, it's not enough to have a structure. You have to know how these proteins are working in the cell, where they're localized. And so this was one of our first imaging experiments of R1 beta and R2 beta in the brain. And you can see how differently they're localized. And okay, this one, that's what I thought. It didn't come through. But yeah, you can zoom in. You can use it like a Google map. You can use it like a Google map, and you can hone in on specific individual cells within each of those regions. And you could see R2 beta goes mostly to Golgi, whereas R1 alpha we can see in the brain, a lot of R1 beta actually in the nucleus of cells. So it's really important to see these proteins in the context of their tissues. And again, looking at going from atoms to molecules to communities, to tissues, and to the animal phenotypes. I think we need all of that, and that's why Dario was so good in the beginning of doing the animal models to see, okay, do you recapitulate the phenotype in an animal model? And I think you need all of these to really understand what's going on. And so I wanted to now just do very quickly, what have I learned since 2019? And just a few little lessons. Here's a map of PKA and disease. This was done by Sylvia Gutkind's graduate student. And there's a lot of diseases that are linked either to G-alpha-S, that's Sylvia's expertise, or through PKA. And you can see R1 alpha is a big target for disease mutations. You can see C alpha. There's some mutations associated with C alpha. And then there's C beta. What is C beta? And so here you can see the mutations, the R1 alpha mutations, carne complex disease, acrocystosis. I told you about that. The C alpha subunit, there are only a few. There's a Cushing syndrome there, and they tend to not associate very well with the regulatory subunits. And then there's a few in PKC, the C beta subunit. So what are these? What is C beta doing? And, if you look at the dark kinome, they had this initiative at NIH. You know a lot about many kinases, many structures of many kinases, but there's also a lot at the very bottom of this kinome where they probably are very important, but you don't know anything about them. And here's C beta. It's down near the very bottom of this. And, how can this be? PKA is kind of the poster child of the kinase family. And so if you look at the expression, RNA expression, you can see C beta, like the R1 beta and the R2 beta, is very enriched in neuronal tissues. And so we asked, is it functionally non-redundant with C alpha? Is it biochemically different? Is it targeted differently? And is it relevant for disease? And so one of my last trips to Europe in November of 2019 was to Madrid. And I visited Victor Ruiz, who had identified these mutations. There was the same mutation in 3 different patients in C alpha, but 4 different mutations in C beta. And they had a very interesting phenotype. They had this polydactyl phenotype, extra digits, and all of these mutations inhibited sonic hedgehog signaling. So this is, yes, C beta is doing something important, and it's different from C alpha. And you can see these mutations there's C beta mutations. One is here, and the C alpha mutation is in this helix binding region where PKI binds. But there's two in this histidine that comes down and interacts with the phosphate on the activation loop, a histidine to arginine and then a histidine to an asparagine, and one here in the glycine-rich loop. So these, we think, are more regulatory kinds of phenotypes, and we really want to explore more what those are. But sonic hedgehog signaling, so I didn't know much about it. It's very sensitive. Like I told you, PKA, a little bit, too little activity, you get these birth defects, like extra digits. Too much activity, then that results in various cancers, like glioblastoma, I think, is one. So what is going on? Smoothen. So smoothen is a GPR that's associated with sonic hedgehog signaling. Usually it's turned off by this other membrane protein patch, and that patch is activated by, then PKA is active in this resting state. So hedgehog turns on patch, and then that activates smoothen. And so once smoothen is activated, glee can now be not phosphorylated and transferred into the nucleus to turn on gene transcription. So what's going on with PKC? And they've really tried to find the G protein coupled GS, GI, what's going on, and they really have not been able to do that. And so March of 2020, right after the pandemic, March or April, Ben Myers from Utah calls me and he said, oh, we have this interesting observation of PKA and sonic hedgehog signaling. And so I thought, oh, he can explain this. So Ben works on smoothen. And so here's a structure of smoothen. But what's missing from this is the C-terminal tail. You cut it off almost all of the receptor structures that are known. And there are many of these 7 transmembrane GPCR structures now known. Cut off the tail because it's intrinsically disordered. And so what we found with Ben, when we looked at this more closely, you could see that embedded in the C-terminal tail of smoothen was a PKI-like sequence. So it has an alanine instead of a serine or a threonine. It has features that resemble the PKI sequence, and when you mutate those residues, it no longer binds to this catalytic subunit. So you have here a cyclic AMP independent mechanism for inhibiting PKA, very non-canonical. And so we're going further, hoping to solve the structure of this complex, and that's been a really nice collaboration. I didn't even know Ben like 2 years ago. So C-beta, just tell you a little bit about C-beta. C-beta-1 and C-alpha-1 are the same size. They have 25 amino acids different altogether, and but there's splice variants as well. So C-beta-2, C-beta-3, C-beta-4, 4AB, a lot of splice variants just at the first exon. And these are the same length, but the others, and they're both myristylated, but the others are not myristylated and they're different lengths. And so if you look at this, you could see those mutations are really enriched in the N-terminal tail and in the C-terminal tail. And there's not at the active site. So these are mostly regulatory differences. And I'm predicting that these are going to be regulated in a different way by PDK1, and that maybe you even have alpha-beta, C-alpha, C-beta dimers as part of the process. So if you just look at expression, you can see like R1 alpha, C alpha are expressed constitutively, whereas if you look at C beta, it's highly enriched in the brain, and this is PKI right here too. It's also enriched in the brain. So some really interesting questions to look at. And then the other thing about the C beta subunits is that they're expressed in this highly tissue-specific manner, as you can see here, but also here. And like C-beta-2, it's the longest one, and it's highly expressed in lymphoid tissues, and T cells, B cells, spleen, highly expressed there. Whereas the C-beta-4 is almost exclusively expressed in brain, and we think in neuronal tissues. And so one of the things that we did as, you know, just to see, can we use the retina as a window into the brain? And this is work with Dorota Skaranska-Krasik, who's now at Irvine. And we did imaging of C-alpha. This is C-alpha in the retina, and you can look at, these are the rod and cone cells here. These are the interstitial cells, and these are the retinal ganglia cells down here. So you can see that's C alpha. And if you look at C beta, you could see, first of all, that it's different. Especially we focused initially on the rod and cone cells, that it's different. And so if you look up close at this, you think, okay, what are those? And so these here are the cilia transition zones. So these outer segments of the rod and cone cells are highly differentiated cilia. So this is this transition zone, which is one thing I'm really interested to find out about. These cone cells have these ellipsoid, these really large ellipsoid kind of structures. And so what are those? So you look at, because the antibodies are incompatible, this is just doing R2 alpha and C beta, but C alpha is pretty much like R2 alpha. It's all in the cell body. It's not in these ellipsoid structures here, whereas that's where PKC beta is really enriched. There's a little bit of it here, but it's really enriched there. And what are those? Those are the mitochondria. And you can just see this is cytochrome C oxidase complex 4 marker that we've just shown to emphasize it goes together with the mitochondria. And you can do the Z-stacks, and it's going right through. So it's really pretty interesting. And I think it opens up a pathway now. We're beginning to look at Alzheimer's disease brains to see is C beta also in neurons, and it is. Can we profile that? Aging, fasting and feeding. There's a lot of things that you can do to just see how do these change as a function of physiological state and or disease. And then I'm just going to tell you this last, I'll do this really fast because I know I don't have much time left, but LRRK2 is another kinase that we started looking at like just fairly recently, 2016, 2017. It's huge. It's 2,500 amino acids. It's got like a kinase domain, but it also has a GTPase domain. So for me, to trap actually crosstalk between a kinase domain and a GTPase domain in the same polypeptide chain is pretty interesting. Lots of crosstalk, RAS-RAF we've heard about, but here they're in the same polypeptide chain. And some of the structure, we got funding from Michael J. Fox and put together this consortium team which was Elizabeth Villa and Andreas Leschner, Fritz Herberg in Germany. And nothing was known about this kinase. And, you know, it was just discovered as a risk factor for Parkinson's disease in 2004. And imaging was done here to show it wraps around microtubules, most likely some of the PD mutations. But we came out in 2020, in the summer of 2020, two structures, one from Andreas of the RCKW, the 4 C-terminal domains, and one of Elizabeth Villa, where it shows the active like dimer polymerizing and wrapping in a helical array around microtubules. And then we had two structures, like one that came out in 2021, 22, the structure of Jisun. This is 2020, the first one. It's the inactive, full-length inactive kinase. This is at 3.1 angstrom resolution. This, Andreas, is at 3.5. So this is amazing for the kinase field. Everybody just looks at the kinase domain because then you can get all the kinase inhibitors. But this is amazing to have this kind of structural information on such a large kinase as this. And I want to just tell you one thing. So this is like the full-length one. This is Jisun's inactive one. And what we could do with like Andy McCammon that has developed Gaussian MD simulation. So you can take your static crystal structure, your static cryo-EM structure, and create a dynamic portrait of it. And so we did that. And so what's interesting about this full-length protein is it has one ATP bound to it and one GTP bound to it. And you can see that's the full-length structure. And there's a G loop that has an ATP bound to it and a P loop in the GTPase domain that has GDP bound to it. And I just wanted to show you the structures of these two. This is the P loop, which I told you binds to the phosphate. And you can see how stably anchored the phosphate is there. This guanine ring can move around, can move there or there, it moves around, but the phosphates don't move. The phosphates are very stabilized by that glycine-rich loop. That's the P loop. Whereas if you contrast that to the ATP binding to the kinase domain, you see it's the adenine ring that's very stable, and the phosphorylates that are now being exposed to a large space. And I think, you know, when you look at a kinase like LRRK2 or SRC, a non-AGC kinase, where you have to, first step is cis autophosphorylation, that you can see there's a lot of autophosphorylation sites in LRRK, putative autophosphorylation sites, and most of them come in this space that is now explored by this phosphate. And I think to bind with a 25 micromolar KD, or KM, which is what you need for KM, for catalysis, the adenine ring is just fine. And for most of these kinases, you don't need a C-terminal tail or anything like that. And so I just want to acknowledge, I've tried to go through and do this as I spoke, of all the people. I've obviously have very interdisciplinary people I've worked with over the years to understand not only the high resolution structures, which is where we're more focused, but also the dynamics, and also what are these proteins doing in cells and tissues, and kind of why I think it's so important now to do the imaging, which we spoke about earlier. So thank you for your attention.
Thank you very much, Susan,
That was a quick
for your
overview.
impressive talk. Does anybody have questions?
Leonard.
Beautiful stuff, Susan. Maybe I can bring this now back to PDK1, since you mentioned the requirement for, or that PKA is synthesized in a fully phosphorylated state, but that's kept inactive by the regulatory subunits. So that implies that if PDK1 is the kinase for PKA, that this happens almost co-translationally or something. What can you tell us about how it works, and is that a growth factor independent function of PDK1? How is PDK1 and PKA brought together for a specific reaction? Yeah.
I mean, I'll tell you about that tomorrow, because I'm going to talk about PDK1 and activation of PKA. How do you do that initial activation step? I think, I mean, from some of our earlier work, and Dario, maybe just, I mean, other people can disagree, because I'm not a biologist. I'm not a real cell biologist like you guys are, but that first initial site that is it's a cis autophosphorylation site on this. It's in the turn region between, and we don't have PKA and PKG. They end with FXXF. They don't have anything after that. No other phosphorylation site. So the first one happens. I think that's pretty easy. It can happen as a cis autophosphorylation by this mechanism that I told you. Then the next step, I think, is you can do two things in PKA. It can autophosphorylate because it has an ARG-XX threonine at the activation loop. If you take away that arginine, it can't autophosphorylate. Then in the cell, it's absolutely dependent on PDK1. And so then PDK1 binds, and I believe you form a dimer, some kind of PK C-alpha dimer or C-alpha C-beta dimer, and that then you have a trans autophosphorylation by PDK1. That's how I envision it, but I don't know. But I think that happens off of the ribosome. And I think once you're folding up on the ribosome, once you get past that halfway through the C-terminal tail, now it's capable of binding ATP. Until then, it can't bind ATP at all. So that little piece, which is pretty much the same in all the AGC kinases, we don't really know what it does. It goes at PXXP, which is stuck onto the C-lobe, and this little loop, that's the same in all of those, almost all of them. Alexander's going to tell us about another helix that's in a few of the PKCs. So somehow that is highly regulated. And it's happening at the ribosome. What else is there with it? I don't know. How C-beta contributes, whether that was a factor in the early studies that Dario did that said PDK1 probably wasn't important for PKA. I do believe that autophosphorylation is important, but probably later on in oxidation, when the holoenzyme is assembled, the cysteine, it gets oxidized, and then it gets dephosphorylated, and then it gets rephosphorylated there. There, I think autophosphorylation is probably important, but when it's being made, I think when it comes off the ribosome, you have that first autophosphorylation. That provides a docking site for PDK1. That's how I think about it, but it could be different.
So I thought that last observation with the different dynamics of the ATP in the P loop versus G loop was pretty fascinating. Is that conserved then for all P loop or G loop containing proteins? If you do similar simulations, they all show that property.
Yep.
And then what are the implications of that for phosphoryl transfer? I mean, if you have a gamma phosphate that's waving around, how does that get stabilized in a coordination that is able to then transfer to a substrate? Is it stabilized upon substrate binding, perhaps?
Well, I think if you, again, I'm just going back and rethinking a lot of these things myself. I think that initial cis autophosphorylation is just a proximity. If it gets close, it will transfer that phosphate to, it won't be a tight Michaelis-Menten kind of complex. I don't think Michaelis-Menten kinetics even works in cells at all. It's protein-protein interactions. But I think that it can hit that. If it gets close, it'll transfer the phosphate. It could be slow, but I think the cis autophosphorylation is easy to see. And I think some of the studies now in SARC, you're seeing that initial cis phosphorylation, autophosphorylation of the activation loop. Then the next step, where you actually have to take, and that'll bring these lysine, aspartic acid residues together. The next step of putting the activation loop in an extended out conformation, that takes a longer time. And I'm thinking what you need that for is for transphosphorylation of a heterologous substrate. And that you have to, sometimes, like when you take SARC to the membrane, it's going to bind to its membrane-bound substrate, and the residues that face out are going to help recognize a substrate. So I think you have to think about cis autophosphorylation versus transphosphorylation of a heterologous substrate, and that is a single protein-protein. It's not like you do Michaelis-Menten kinetics where you have a huge excess of the substrate peptide. It's a protein-protein thing, and then you have factors of what makes the protein come there. You recruit it to the membrane, you can phosphorylate, but it's not going to be turning over a lot, I don't think. That's kind of how I'm coming to think about it.
Thank you very much. Sandra, maybe you can start preparing your presentation, and we have another question there. And here first.